Thanks very much. You'll see my Twitter feed up here and the Twitter feed of the museum. And actually, I tweeted before, the question that we were asked is can we stop the next extinction? And my answer to that is we may be able to, and it may be the trees that help us. Okay, so trees obviously take the carbon dioxide, which is responsible for climate change, out of the atmosphere, but we don't really understand how that elevated CO2 that they're currently putting in, we're currently putting into the atmosphere, we don't understand what's going to happen to that. So maybe trees will save us, but we need to understand more about how those trees will respond to the climate change that's happening now. They could be our saviors. We all know that climate change is happening. This was from the BBC website recently, as well as the elevated CO2, that there are knock-on consequences to that, such as rainfall patterns and elevated temperature and so on. I did also mention in my tweet that I was going to explain how trees were going to do this using gin and parsley. I do have some gin and I do have some parsley. And I will explain in a little bit more detail that maybe at the interval if you're interested. We have also in the interim been experimenting with what we now think are very exclusive botanical cocktails, which actually do underpin this. This is an extract of parsley, which I bought from the Lidl not 2 hours ago, 3 hours ago, and the gin, which I bought from Tesco Express. Other supermarkets are available. If you grind up a happy plant and you illuminate it with the right sort of light, and this is a blacklight torch, a UV torch, not a normal torch, and that has the wavelength that plants want to do photosynthesis. This is under ordinary light, where you can see the chlorophyll. This is under what we call a black light, and it fluoresces red. What's happening is, because of the gin is an alcohol, it's dissolved all the membranes, the chlorophyll has come out, but it's still biologically active. It captures the light, which it normally would take that light, make an electron, and that electron flows through the membranes like electricity and provides the energy to take that carbon dioxide and make it into sugar. When it's in this solution, it can't do that because you've dissolved the membranes in the gin, and it emits the same energy that it would have used to do photosynthesis. It emits that energy as red light. So a subcellular activity there that we're going to think about in a global context. Here we can see that fluorescence in a single cell, we can see it in a single leaf, and we can even see it from a satellite. So this is a global phenomena happening at a subcellular level. If you go onto Google Earth, actually let's just do that now, and I'll just go on the internet and show you a Google Maps. And hopefully if I've centered this right, if we zoom in, a place called Norbury Junction, just north of Birmingham, you see this piece of woodland. This is the Grand Union Canal, and there's this woodland here. So we've gone from the subcellular to the global, and now we're zooming in. If we zoom right in, oops, I'm going the wrong way. See this bit of woodland? You might notice something slightly strange about this woodland. It has 6 enormous rings of pylons in it. And that is one of the biggest experiments in the biological world. It's called a FACE experiment, free air carbon dioxide enrichment. What we want to understand is what that process that's happening in the gin-extracted chlorophyll, what's that process happening in a natural oak forest? 3 of these rings have elevated CO2 pumped into them, and 3 of them have ordinary fresh air. And over the next 10 years, we're measuring everything that goes on in that ring, from the photosynthesis right through to how the insects respond and how the soil responds. In order to really understand what is going to happen as the CO2 goes up, the most rapidly the CO2 has ever gone up, we need to understand where plants have come from. Hence my question, plants have never properly colonized the land. Plants started out as algae living in the sea. When you live in the sea and you float around, you don't need to worry about getting your CO2 because it's dissolved in the water, and you don't need to worry about water because you live in it. As soon as you colonize the land, you start to get some problems. The early land was a very harsh environment. There's a problem with getting enough nutrients, but we'll leave that aside for now. But it's actually very dry. It's actually, losing water becomes a big problem. If you lose water and you're a plant, you wilt. Right, so what are you going to do to stop losing water? You have a cuticle. You have a waxy coat, like we would put a mac on when we go out in the rain. That's fine. Where you can see this is a cabbage with a waxy coat, and you can see it's stopping the water getting in because of the wax, and you can see it here being pulled off. It also stops the water coming out. We now, as a plant, have a fundamental problem. We've colonized the land, we don't lose any water, but we can't eat. We can't photosynthesize. We can't take any CO2 up. So how are we going to solve that problem? We solve it with this, stomata. This is a pore that plants have to effectively breathe through. So now we've got a cuticle, but we've got holes in it that can take in the CO2. That's brilliant, all sorted. Or is it? Because actually, if the CO2 goes in, the water can sneak out. So we still haven't solved the problem properly. If we look along the evolutionary sequence of plants as they did colonize the land, if we look at mosses and liverworts, they have this pore. There's the cuticle, and there's the pore. Great. It's okay, but CO2 goes in, but water can come out. And effectively, as the water comes out, these are the cells on the leaf. This is the stoma, the hole. This is the pore. And there's the water coming out, but it's just a hole. And as the water comes out, the leaf will dry out. So that's not good. That's not good for the plant. You've still lost your water, even though you've regulated it a little bit. So what's the next stage that actually we get? What's the next stage of control? Well, if you look at a fern like bracken, this is bracken, they also have nice stomata, and those stomata are a bit cleverer. They're a bit more sophisticated. Also point out that a fern still lives in wet ground because they still need to have the sperm of fern they look like animal sperm, and they still need to swim through a film of water. So they still haven't escaped water for their reproduction, but they're starting to control the loss of water with these stomata. And these stomata that ferns have can close. Okay, so there's the stomata. You start to lose water, and the hole actually closes, but you're still losing water because the stomata close actually. And I'll just show you this. This is actually my wife's Brompton inner tube. So hopefully on the way home she didn't get a puncture, or she'll be very cross with me. These stomatal cells are actually composed of two cells, and when they're under pressure, the cells actually separate. And they're under pressure because water moves into these cells by osmosis and generates a huge pressure. The pressures in these are about 6 atmospheres, not in this because this is an inner tube, but in the actual cells about 6 atmospheres, about 2 or 3 times what you get in a car tire pressure. And as the water leaves, because the plant is droughted, the stomata will actually close. Close, go on. You say you should talk to your plants. There you go, look, it's wilted and it's closed. That is a successful science demonstration, okay? So, but they've still lost water because they've had to wait for the water to leave the leaf in order for the stomata to close and start to regulate it. But it's better than being a bryophyte. The other thing that ferns have done, which gets away from the water, is now their seeds and their pollen are dry. So they've escaped the need for water for reproduction. Sorry, not the ferns. This is the next stage. These are the cycads. And these are called gymnosperms. Their seeds are naked. And the name comes from, if you ever go to the gym, anybody go to the gymnasium? Hopefully you go naked to the gymnasium because that's why it's called a gymnasium, because the Greeks went to the gym naked. Gymno is the naked root. And the seeds of a pine tree or a cycad like this don't have a cover, so gymnosperm, naked seed. Cycads are what Dippy would have ate. Okay, cycads were around at the time Dippy was, and this is what Dippy would have ate. So they've escaped the need for reproduction, for water for reproduction. They don't have sperm, they now have dry pollen. But now their stomata have got an even cleverer system. So before the leaf dries out, those cells in the stomata get a signal from the roots. The roots detect that the soil is drying out, and the stomata closes without the leaf drying out. So there's a root-to-shoot signal in the plant. It's a hormone called abscisic acid. So the stomata shuts before the leaf dries out. Actually, really sophisticated control system. Flowering plants, angiosperms, angiosperm, angio meaning container. So now there's a nut. Here's your seed, your naked seed, but in an angiosperm flowering plant, they're contained in the seed, and the seeds can dry out and don't have to have water. They also have the same type of control system. So CO2 goes into the leaf and makes sugar, and here you can see, for example, tree rings. The better season you have as a tree, the bigger the tree rings. So stomata, the CO2 goes in. You've still got the problem with water coming out, but you've got this control system. Let's think about how those evolved and what those evolved in. Similar to the previous talk, here we've got plants colonizing the land, and here we've got the present day, and here we've got the CO2 levels. We now know CO2 levels about 300-400 parts per million. They were orders of magnitude higher than that at different times under evolution of plants. So ferns had a higher one there. Cycads evolved here. The little red bar is when Dippy was around. And then angiosperms evolved here. So our plants today are dealing with the lowest carbon dioxide they've ever had to do. And so the interesting question is, how have all these control systems, how are they going to respond to these elevated CO2s? The CO2 is now going up faster than it's ever done before. So in 100 years, it could go up to 1,000 parts per million. Absolutely huge rise. So how are these plants going to keep track of that? And that's what these face experiments that we're doing are for. If you give plants more CO2, they make more sugar and they do better. Yes, that's true. It can happen under certain circumstances. These plants are grown under elevated CO2 and these are grown under ambient. However, if I said to you, I'm going to give you more food, but it's just going to be sugar, you wouldn't consider that to be a balanced diet. Like you, a plant can't just eat a bag of sugar. It needs all the other nutrients as well. So it's much more complicated than just growing bigger. So that's what the face experiment is going to tell us. 3 of those rings are going to have elevated CO2 and 3 don't. Here's the size of this thing. Absolutely huge. There's you see the scale of it with people. Indeed, the pylons were put in by helicopter, so it didn't disturb the ecosystem. Prince Charles came. Prince Charles refused to put a helmet on. Just think if a branch had fallen on his head, how that would have affected the succession. We get live data feeds from there, and that's going to go into schools and stuff. The question is, if we give the plant more CO2 and it makes more sugar, maybe it'll be able to defend itself against pests better. But maybe if the plant doesn't get other nutrients, the nutrients are diluted and the pests will eat more of the plant. So we don't know with those conflicting hypotheses what will happen. Plants squirt loads of carbon into the soil to stimulate the growth of bacteria and fungi, which are symbiotic with them. And mushrooms are the fruiting bodies of those. We don't know what's going to happen with those. So there's all sorts of questions as to what's going to happen to that CO2. Will it affect this stomatal regulation that we've talked about over evolutionary time? Will it go into the soil? Will it affect defense? Will it affect their reproduction? So you can see that stomata can affect that regulation of water and carbon dioxide over minutes, but also you can put fewer or more stomata into the leaves over decades. If you look back at herbaria specimens in museums over the last 300 years, the stomatal density has gone down. As the CO2 levels have gone up, plants have already started adapting to this by putting fewer stomata into the leaf so they lose less water because there's more carbon dioxide about. This is early data. I think this is one of the first public showings of this data. This is the first year data from a PhD student of mine, Anna Gardner. And this graph nicely encapsulates the stomatal conductance. So these are the stomata are really wide open, these are really closed, and this is the rate of photosynthesis. The red dots are the ambient rings, the normal carbon dioxide concentration, and the green dots are the elevated CO2. And there are two things to take from this that hopefully tie in at the end of this talk from the evolutionary perspective of the balance between getting more carbon and preventing water loss. The first is, you can see the blue dots tend to be higher than the red dots, so that shows you that photosynthesis is indeed higher at elevated CO2, so that's one result that's coming in. But also there's a stomatal effect, because the stomata, the blue stomata, the elevated CO2, they're more closed. So not only is the plant taking in more sugar, it's also trying to save water as well. So two things starting to come out of this data. I'll just finish with this last slide. I'd just like you to reflect a little bit that I showed you subcellular stuff with the gin right at the beginning, and we're talking about global events that are going to affect everybody over the next decades. And those global events, those global warming climate change events and the impacts on plants and ecosystems and animals and so on are actually being mediated through this tiny little cell. This is where the battle for climate change is actually being fought. And the responses of those plants and how they will do it will tell us whether we are going to have another extinction or not. Very much.
Thank you, Jeremy. Does anyone want to kick off the questions? Put your hand up if you are ready to ask a question. It's David's only job here tonight to run after you. So anybody has a question, stick your hand up. Maybe you can answer one of mine first, Jeremy. I was very impressed actually at the start with the gin and the black light, nearly making plants a wee bit sexy. Then you ruined it with the whole put on a Mac when you go outside. You showed us there the face experiments, ones that are happening in England. Are there other ones happening around the world, or are there plans for other ones around the world?
Yeah, there are a number, because obviously the one that we've got in Birmingham is an oak ecosystem. It's obviously a western temperate forest, which is quite iconic. There's also one being developed in the Amazon rainforest, which is clearly one of the biggest sinks for carbon. There's also one being talked about up in Sweden on the tundra in Sweden, and the one that's really coming to an end now but has generated a lot of data is in Australia in the eucalyptus forests just outside Sydney.
Okay. Yes, we have a question here. You go ahead.
I take it you can measure stomatas, and you would notice there's more stomatas happening.
You can measure their aperture individually. So when I put the slide up of the over minutes or over millennia, yeah, and you can measure their density simply by counting them over a particular area. So those are the two ways that you can regulate CO2 input and water loss.
Are plants in the process of creating more stomatas now?
They're actually putting fewer in. So for example, there's an experiment you can do. It's quite interesting. If you take a plant and you put a cuvette on one of the leaves and you give that leaf more carbon dioxide, and then you count the stomata on a new leaf that's not got elevated CO2, that has fewer stomata because the plant realizes it's got more carbon, so its priority then becomes to lose less water.
So there's less carbon monoxide in the atmosphere today?
There's less than there has been in evolutionary time, but it's starting to go back up. So the museum data suggests that as carbon dioxide has gone up over the industrial period, plants are putting fewer stomata into their leaves. So their priority is, once they've got enough carbon, their priority becomes saving water.
So you can prove it.
Who?
Climate change is happening then?
Climate change is happening, yeah. It's in the museums. I think there's a question at the back as well. I think.
Yeah, we've got a couple of questions. See, when he said Donald Trump, everybody put their hands up. Everyone was like, we can all prove him wrong. It doesn't take Jeremy. Yes, there's a question at the back. Go ahead, please.
Hi there. I'm basically just wondering whether we have enough information from what the plant equivalent of the fossil record would be as to how different species of plants might combat mass extinctions and so forth, how they would come back from that.
how they combat the mass extinctions. I don't know on the species level. I mean, maybe the other people can comment on that. But in terms of the types, those classes of plants, so mosses and so on. So you've got mosses and bryophytes, then you've got the ferns, then you've got the gymnosperms, and then you've got the angiosperms. Each of those has evolved in sequence, but all of those are here today. So as broad groups, none of them have gone extinct, like, say, all the dinosaurs went extinct. The thing that I put up at the beginning when I showed this sort of faked up picture of the harsh landscape, things like those plants, which one of them was called Cooksonia, which we only know from fossils, those very, very early plants, we haven't got any examples of those anymore.
We have another question here, David, kind of, if you run really quick down the side and then into the middle. Keep your hand up just so he can see you. There. Thank you.
About the use of herbarium specimens, were you able to find statistically sufficient numbers of the same species from the same locality in herbarium material?
It's not my data, unfortunately, but it's in a Nature paper, so I trust it. And I think the stats is good. And the thing about herbaria is that our predecessors were incredibly good at collecting and recording where they collected things, particularly in the botanical field. So I think we can be, even though they didn't have GPS and all these other things, I think we can be reasonably confident. The face experiment, though, is doing that over a decade. So we have all those leaves in storage. And over the next 10 years, we will be comparing stomatal densities in the rings and not in the rings. And we can have a really tight statistical control over that, but in a natural environment. That's the key thing. It's not in a lab, it's not artificial, it's just a forest.
Do we have another? Yes, there's another question, David, just down, back the way you're coming there. This is really fun, David.
Thank you. I really enjoyed your talk. I was just wondering if scientists will use this information to build future technologies or future sort of mass planting of trees or something like that with a particular stomata density.
I think that's right. I mean, I think we have lots of models of climate change and how plants would respond to CO2, but to use a horrible word, they're not very well parameterized. We don't have the numbers to put into those models, and what we're doing here is putting the numbers in, but you're quite right. If we can understand which trees capture that carbon the best, then those are the ones that we should be planting. For example, eucalyptus from the Australian face that I mentioned, if you give young eucalyptus trees more carbon dioxide, they grow really fast because they use that food because they've got enough nutrients. If you give older eucalyptus trees more carbon dioxide, they don't. They're more interested in pests and not losing water. So looking at oaks, maybe we see oaks which are much longer lasting and have a lot more biomass, not more weight, not more sugar. Maybe they'll be the ones that we should be planting to capture that carbon dioxide. The other thing is, of course, that they capture more carbon if you feed them more than just sugar. So if you fertilize plants and give them more carbon dioxide, they will do a lot better. So everybody who's had a tomato sandwich today, those tomatoes, if they're greenhouse grown, they will have been grown under elevated CO2 because they use the heaters from the greenhouse. They put the CO2 from the things that heat the greenhouse back into the greenhouse. But that CO2 is used by the plants because they're fed and watered artificially. So you've probably all eaten plants that have been grown under climate change.
Do we have any more questions? Yes, question over here. Oh, we've got two. These could be our last two questions here, David. Just here at the front.
I am. Considering the number of trees that have been chopped down around the world lately, or over the last 5 years, what correlation is that with climate change?
That's a good question, and we probably don't know. I think in terms of, I mean, if you think of an oak tree, it lives for 600 years, and so that's longer than we've looking back over the Industrial Revolution. So I think how they react in the next few hundred years will start to tell us that, and the same with the Amazon. I think chopping down trees, it's more the coal that I think is really giving us the acceleration of climate change. The other big thing that we don't know a lot about is the sea, because of course 2/3 of the Earth is the sea, and the algae in the sea also do the photosynthesis. One of the slides I showed was algal chlorophyll fluorescence. If those algae are stimulated by the elevated CO2 and take that up, and if they die and fall into an Atlantic trench and are out of the system, then that will be really helpful. But if they keep recycling the carbon and putting it back in the atmosphere, then it won't. So that's a long-winded way of saying I don't know the answer to your question.
It sounded impressive, though. Thank you for that question. That was great. We're going to take our last question here. No pressure. Go ahead. Make it a good one.
I just noticed you were saying that Dippy was, like dinosaurs were feeding on cycads, which is kind of heartbreaking because I always imagined that dinosaurs lived in forests of trees. But if that were true, I mean, are all cycads small or were they like tree size at the time?
Huge,
or? Yeah?
huge. I mean, nature has a way of filling a niche. So, you had cycads and they were massive trees, massive ferns like the tree ferns that we see in New Zealand now. That's pretty well all that was left, but they were massive things. When the climate changed and they weren't able to do as well because they weren't as well adapted to the changing environmental conditions, then angiosperms with their dry pollen and their seeds and their much more sophisticated control of water loss were able to outcompete them. But an oak tree fills the same niche and is pretty much the same size as one of those ginkgos, cycads would have been. Incidentally, Dippy wouldn't have chewed them. Dippy used to apparently shear off the plant and then just swallow them whole, and they found stones in the stomach that would grind it up and then it would ferment rather than... So the skull didn't need to be big and chew, apparently. And I speak as a plant biologist, so don't take me on that.
Some people still eat like that today, actually. Can we please get one more round of applause for Jeremy Pritchard? Thank you, Jeremy. Thank you.